So, getting down to things that are testable. Two major classes are cells and the brain. One class are neurons, the class is glia. You know that, right? Okay. So, what is the purpose or function of neurons? And remember the two classes of questions you could ask: mechanism of function, mechanism of function. Okay. So the function of neurons is to receive, integrate, transmit information. Neurons themselves. It's been recently determined that an average, or number, that on average the human brain has 86 billion neurons. Now, when I was your age, sitting in class, I was told that there were 10 billion neurons in the human brain. You've got a lot smarter since then. Last year in this class, the number that was given was 100 billion. Dumbed down a little bit since then. Okay? So what happened that we lost these 14 billion neurons? Well, the 100 billion was a rough calculation that was based on many different areas of the brain and a very small section of microscopic sections, counting the number of neurons in there and averaging those together for different kinds of brain tissue and going, that number was used for decades. Okay? What happened a couple of years ago is that you're never responsible for people's names, right? I have to mention it's about the brain and physiology. It's all I really care about. But a South American scientist actually took human brains and disaggregated them, dissolved them, so that the nuclei of neurons could be counted, the cell counter, did that sample size, and from that, in multiple human brains, came up with an average of 86 billion. So that's actually a counting. It's much closer to the real number. Okay? So you think that would have been known a long time ago. That's actually, okay? So we lost 14 billion neurons. But, oh well, at least we have good information now. And importantly, it's been calculated, although this is a rough number, that neurons receive an average of 5,000 synaptic inputs. Right? And each synaptic input is actually a processing unit. So if you take those two numbers together, that 86 billion neurons with an average of 5,000 synaptic inputs per neuron, that's more synapses in your brain than there are stars in the Milky Way galaxy. It's a big number. And 86 billion is an unimaginably large number. And you have the 5,000 or so synaptic connections. That's why you get an A in the course. There's some complexity. It's an enormous amount of processing power that gives you the synaptic hardware you need to get an A in the course. You've all got it. Okay. There are the other kinds of cell in the brain, the one that I'm almost going to ignore in this course. There's the glia. The term means glue in Greek. Not so good at the ancient language. It's part of it. It could be Latin. Don't really care. And that was because of the original conceptualization of what the glia did was just this kind of structural support for the all-important neurons. It's just, they filled in the space. We now know that glia have much more important functions than that, and actually in some types of glia are involved in modifying the processing, the information processing by neurons, and some glia actually do some information processing themselves. Diaco and the Department of Cell Biology and Neuroscience, for example, from the UCR scientists making discoveries about glia and their processing power. Okay? There are roughly 10 times more glia than there are neurons. So 86 billion times 10, approximately. Right? There's actually more of them. And there's three major types of glia. There are the oligodendrocytes and Schwann cells. And these are basically functionally analogous types of glia in different parts of the nervous system. So the oligodendrocytes are in the central nervous system, so brain and spinal cord. The Schwann cells are in the peripheral nervous system, basically in our And what they do is analogous to each other. Basically, they will wrap around and insulate the axon. Okay. There's the oligodendrocytes. See some over. I can't point to this. Over here, there's some oligodendrocytes. I'll be prepared. Right. So this is an axon of a neuron. And we're going to say oligodendrocytes right here. So there's one, there's another, there's another, there's another. There's a bunch of different oligodendrocytes. And what they do is that they, the oligodendrocytes will wrap around the axon. If my arm is an axon, they will wrap around the axon and insulate it, thereby improving the ability to transmit information long distances in ways that we'll get into in some detail later in the course. Okay. Another kind of glia are the astrocytes. And astrocytes do lots of different things. They provide supporting functions for neurons. They buffer extracellular concentrations of ions, which turns out to be very important for the way that neurons generate electrical signaling. So that's actually important in information processing. They're a major source of metabolic fuels for transport of metabolic fuels for neurons, and that's important because the brain is the most energetically active tissue in the body by far. Cell per cell, neurons use more energy than any other cell in the body. Okay, by a long shot. And in fact, even though the brain in a typical human may only be 2% of the body, they actually use 20-25% of all the calories you eat every day. Like a neuron does. Okay. And as I said before, they also affect information processing by neurons and do some on their own. And in the microglia, they're basically phagocytes that clean up cellular debris and some other things I'm not going to get into. Okay. So let's now go back to neurons and get into some detail with the morphology of neurons and what they look like in the different parts. Okay? So, same diagram, whichever one you're most interested in looking at. Okay? So, this is my attempt to draw parts of the neuron. I'm not an artist, but hopefully this will work. Okay? So, here, this is one neuron here. And this is part of another neuron. And so here we're interested in the parts. So one part common to neurons is the cell body. The cell body is the part that surrounds the nucleus. Okay. And from the soma, from the cell body, you have two different kinds of processes. Now the neurons are cells in many ways like other cells, and they have all the typical cellular, organelles and the cell membrane and so forth. But they're specialized for information processing. So that's where we're going now. Okay, so one type of process that comes from the cell body are the dendrites right here. And this particular diagram illustrates 4 dendrites, right? This long paper structure. Functionally, the important element of dendrites is that they're a main source, or like antenna, if you will. They're the recipients of information. Most synaptic inputs come to dendrites in the cell body. Okay? So most information is received by dendrites in the cell body. More on that later. The other kind of process that emanates from the cell body is the axon, right here. And the distinction is that while there may be many dendrites, there's only one axon actually exiting from the cell body. For some neurons, it begins with a structure called the axon hillock. So this little thing right here is depicting an axon hillock, right? Literally little hillock. Okay. Some neurons have axon hillock, some do not. Okay. And the distinction, one of the distinctions of the axon from the dendrites is that where the dendrites are typically tapered, and they have a bunch of different shapes, the axons typically are fairly constant in diameter along their entire length. Okay. The axons will frequently branch. Okay, so you see a single branch here, it's called an axon collateral. Don't ask me what the difference is between the main axon and the axon collateral. I'm not going to ask you, and that doesn't matter to debate anyway. Right? So branches called collateral. And importantly, the axons will frequently terminate in what's called a presynaptic terminal or gluton, so it's got lots of different names, okay? And the functional importance there is that's the part of the neuron that most frequently releases transmitter. Some neurons release transmitter from other places. We'll get into exceptions later on. Right now, I'm going to classical neurons. So we have a foundation. So going backwards a little bit, so here you have the type of glia called oligodendrocytes, which I touched on earlier. And as I mentioned before, those literally wrap their membranes in a spiral around the axon and improve the ability of the action potential to be conducted down the axon. Okay. Not all neurons have oligodendrocytes. There are some that do them, some that do not. We'll talk about that one later. Okay. And between the oligodendrocytes, you have these gaps here, there's a gap there, there's a gap there, there's a gap there, and so forth. These gaps are called the node of Ranvier. Okay. And those are functionally important in ways that we described later on. Okay. And then going back to the terminal, the zone of output, we'll know about that later too. You have within the presynaptic terminal synaptic vesicles, these little circles here are representing spheres, and those are organelles within the presynaptic terminal that contains molecules of neurotransmitters. Different neurotransmitters will have different number of molecules, but as many as 6,000 molecules per synaptic vesicle. Okay, so lots of molecules per transmitter. Even though these guys are really, really small. You can't see them except in the electron microscope. Okay. And what happens in terms of information processing, when an action potential arrives at a presynaptic terminal, it will cause transmitter release from a subset of the synaptic vesicle. The transmitter will cross the synaptic cleft, that's the gap between neurons, which is actually very tiny, exaggeration of the distance, okay, and will bind to receptors on what's called the postsynaptic density. Okay, so here you have another neuron. So one of the important parts is that information transmission in neurons mostly is polar. It's in this direction, right? From dendrites and cell body down the axon to another neuron. There's some important terminology here, and that is the presynaptic and postsynaptic. So here that is terminology that's with reference to the location of the synaptic cleft. So here's the synaptic cleft. It is that gap between the neurons. It is the gap that the molecules of neurotransmitters cross from the neuron that releases the neurotransmitter to the neuron that has the receptors that the neurotransmitter binds to produce a synaptic potential on another neuron. Okay? So in terms of that, if this is the particular synaptic cleft we're talking about, this is presynaptic and that's postsynaptic. So you see the labeling down here? So this reference to this particular synaptic cleft, this is a postsynaptic neuron, and this is a postsynaptic density with reference to this particular synaptic cleft. This is a presynaptic terminal. Okay? But here, what confuses students is you have to know what the point of reference is. And the point of reference can change. So if I was using a synaptic cleft here as a point of reference, this would be the postsynaptic neuron. So this is presynaptic relative to this synaptic cleft, this neuron would be postsynaptic relative to synaptic cleft. And the terms pre and postsynaptic are used very frequently in neuroscience. Questions? So, a little bit more realistically here, so this is a little bit better attempt than what I did over here. So here we have two neurons, and both of these have cell bodies, there and there. They both have nuclei, there and there. They both have dendrites. Any dendrites here? Okay. Dendrites here. They both have an axon hillock there and there. And they both have axons, in this case with axon collateral, presynaptic terminals here releasing transmitters onto this neuron. And the biggest point I wanted to make with this particular diagram is one I made before, is the major direction of information transmission is shown by the arrow. Okay, so here you have a bunch of axons forming a bunch of different synapses on this particular neuron and its dendrites and cell bodies. Remember, an average of 5,000 synaptic connections per neuron. That's the average. It goes up to 100,000 on some neurons. Average. Okay, so the information is coming in this way. The transmitter release in these synaptic clefts binding to receptors on this neuron will cause synaptic potentials. I'll explain that later. Will cause synaptic potentials. The synaptic potentials are integrated at the axon hillock, and under some conditions, those synaptic potentials will induce the neuron to generate an action potential. The action potential will travel down the axon, cause the release of transmitter, and start the process over and over again. That's the direction of information transmission. Okay. So this one's a slide out of your textbook. And so there are a couple points here. One is that neurons are cells, right? So we've got endothelium, we've got mitochondria, and then a variety of other organelles with cells. Okay? But they're specialized for information transmission. And so here you see a dendrite, there's another dendrite, there's another dendrite, another dendrite. Here's the axon hillock, and there's the axon with the oligodendrocyte. And here you see an input to the dendrite. In fact, there's more complexity than I described to you because some neurons are called synaptic spines. Okay? So there are these little protrusions here on the dendrites. And these are, for many neurons, the point at which excitatory synaptic transmission proceeds. And interestingly and importantly, not necessarily for this course so much, but for other courses, each one of these spines is actually a processing unit that's modifiable. And this is that intra-output relationship I mentioned earlier. Okay, so transmitter being released here onto the synaptic spine, depending on the rate, can actually change the shape of the spine. So you can do video of neurons. You think of them typically as being static, and under certain conditions, rapid input or slow input will cause the synaptic spine to actually change shape or they move around. Okay, so this is processing is happening in the brain right now. Okay. The other general point I want to make out of this slide is organizational. So some people think of the brain, the nervous system, as being like an amorphous block. In fact, the evidence suggests that they're highly organized. You get some of that out of this diagram. Well, you see two neurons that look similar. These are both pyramidal neurons. You will need to know what that means later on. Pyramidal neurons, types of neurons. There are roughly 1,000 different classes of neurons, really original 3. This is one of them. Pyramidal neurons. The pyramidal neurons have this apical dendrite. You don't need to know that name. Apical dendrite. So that's the apical dendrite. And these are oriented similarly in all of the pyramidal neurons in this brain site, the cortex. So the point I'm making is that these neurons are not randomly arrayed, right? They're like little soldiers organized the same way with their apical dendrites going in the same direction. Okay, so there's organization here. And the other point is that there's different cell types in the same brain region, so here's a different kind of neuron here. You can see this kind of neuron doesn't have these synaptic spines, right? So that's just a feature of some type of neuron. So a neuron, not all neurons are created equal. There's a class of neurons. I said earlier there are roughly 1,000 different kinds of neurons in the human brain. And here are 4 types. 3 of them you'll need to know. Okay, so the 3 that you'll need to know are the Purkinje cell here, right? The pyramidal cell here, and the motor neuron here. Okay, so I'll explain more about those later. Sorry for the later stuff. You're probably tired of hearing it, but we'll get there. And I'm laying groundwork for the future. Okay. So all of these neurons have a similarity in that they have nuclei, cell body, dendrites, and axon. Okay. So cell body, dendrite, axon. Cell body, dendrite, axon. Cell body, dendrite, axon. But they look very, very different. And that's a different classes. And they have different functions, and they have different resting membrane potentials, and so on and so on and so forth. So they process information different. Not all neurons are created equal. And also, you get out of this, you see the sort of variety of dendrites, etc. There's also evidence within this picture of the degree of organization. So if you look at the Purkinje neuron, also called the Purkinje cell, right down here. This is 2 different views of the same Purkinje cell. So this is this very complex dendritic tree here from one view, and the same Purkinje neuron from a different view. Now, whereas the average neuron has, 5,000 synaptic inputs, the Purkinje neurons have 100,000 synaptic inputs. Average. And so if you look at the Purkinje cell this way, so if I pretend my hand is a Purkinje neuron, this is the cell body and these are the dendrites, this would be a frontal view of the Purkinje cell in axon. Right? That's a frontal view of the Purkinje cell, that's the profile view. And so these Purkinje cells have a planar dendritic tree. Right? This tree goes out in plane, let's look at it like that. And what that demonstrates is a high degree of organization in the dendritic tree. And it may be the case, that's the point, that's actually the major point. High degree of organization, you can see it in that cell type. It may be that the other cell types have an equivalent degree of organization in their dendritic tree, but it's not visually accessible. Okay? There's more. So if you take one Purkinje neuron like this, where are the adjacent Purkinje neurons? They're all lined up like this with all their dendritic trees in parallel with each other. So this is one Purkinje cell, the next one. Not like that, like that. The next one's like that, the next one's like that, the next one's like that. And then you go down for a different row, they're like this, right? They're like soldiers in formation. Just buried and these Purkinje cells are in this cerebellum, right? So there's very visible high degree of organization of the Purkinje cell as well as the relationship between the Purkinje cells. And there's also a high degree of organization in the input. So, if I'm now pretending that my laser pointer is an axon, the axons to Purkinje cells go through like this, right? So forming a synapse different kind of synapse at each point. So there'd be a synapse here, the next neuron there would be another synapse, another synapse, another synapse. Okay, so there's other kind of synapses that don't terminate. There's called impossible synapses, different forms. The neurons do lots of tricks, right? Change things around in different circumstances. Okay, and so there, what that suggests is that it's timing mechanism, because this neuron is going to receive the synaptic input before this neuron, which is going to receive synaptic input for that neuron. And what is one of the major functions of the cerebellum? It's about motor control. It's about timing and the ability to dance, to do martial arts. Timing is very important in motor control. It's also involved in learning about those things, the cerebellum. And so the suggestion just from the structure is that there's this timing feature it's very evident in the Purkinje cells. But the major point I'm trying to make here, very simple, is that there's a high degree of organization visible in this particular type of neuron, in this particular location, and it's likely that equivalent degree of organization exists in other areas of the brain, although it's not evident. Now, we don't know that. It's just something to think about. Any questions? So, making a point that I made before, human brain, conscious brain, human brain, conscious brain. So now you're looking at sheep brains, why sheep brains are all different than human brains. And human brains are hard to get hold of anyway. But the point is that it's useful because the sheep brain is highly analogous to the human brain, and actually the conscious brain is as well. So if you compare these two brains, and oh, by the way, this is a hemisected human brain, and this is the corpus callosum. That information superhighway I mentioned earlier, the one that if you split it, you end up with people that act like they have two minds inside of one body. Okay. Essentially, virtually every structure that exists in the human brain has an analogous structure in the conscious brain, and vice versa. Every structure, or virtually every structure that exists in the conscious brain also exists in the human brain. What differs between them is the absolute size of the brain. Obviously, human brains are a tad bigger than the conscious brain, and there are more neurons. And then there's also a difference in the relative size of different substructures within the brain. So, for example, humans have the greatest amount of cortex of any species known, right here, whereas the conscious brain is virtually absent. But on the other hand, the conscious brain has a very large optic tectum here, which is analogous to the superior colliculus, which is minuscule in humans, right? So it's a different relative size. Okay? But repeating a point I made before, if you look at the morphology of the neurons within a conscious brain and the human brain, they're identical even to the eye trainer. So the difference in the cognitive ability of those two species is not a function of the complexity, greater complexity of the neurons in the human brain. It's the greater number, the greater interconnections, the greater wiring. Right? And that's why people study the sheep brain. Let's see. topic. We have these different kinds of neurons. 1,000 different classes are on. They're very similar, and they have these similar functional zones. Okay, here. So this is again a symbolic depiction of a neuron. Dendrites, axon hillock, axon presynaptic terminal. So the soma and dendrites are the zone of input of the synaptic input. The axon hillock is the zone of integration, and like I mentioned before, synaptic potentials that are generated by the release of transmitter onto the dendrites and the soma cause synaptic potential, which is electrical change. But later, those electrical changes, synaptic potentials travel towards the axon hillock. They're integrated there. That's why it's called the zone of integration. And under certain conditions, that will generate an action potential beginning at the axon hillock and traveling down, conducted down the axon to the terminal gluton. The axon being the zone of conduction, the action potential is conducted down the axon, or the presynaptic terminal, the zone of output, because that's where the transmitter is released from. And the point here in my last 30 seconds, and I'll come back to this one, it's complicated, is that each one of these zones is functionally different. Has different molecular mechanisms within it. Okay, so I'm not going to finish this one. So these are the zones here. These are the events that happen within it. Synaptic potentials, action potentials of the transmitter. The important point here, the different molecular structures. The zone of input has receptors. The axon hillock, axon, and presynaptic terminal has voltage-gated ion channels, and the ions that are conducted through those different channels are different from each other. So these are different functional zones. And one of the things you want to do that, by me saying it, you won't do sometimes, distinguish synaptic potentials from action potentials. They're different. I will see you next Tuesday. Have fun tomorrow. And I'll have office hours outside.